Today I will be showing the mechanism for the synthesis of hydrobenzoin. I will also discuss the stereochemistry that's involved in this reaction, and I will go over how to characterize hydrobenzoin using proton NMR spectroscopy. Right here I have the reaction scheme for the synthesis. A complication that arises in this reaction is determining the stereoisomer of hydrobenzoin that is produced after we react benzil, this molecule right here, with sodium borohydride. As you can see, hydrobenzoin has two chirality centers right here and right here. What this means is that there are three possible outcomes that can occur after we perform the reduction reaction of benzil. The first outcome is when we only produce the meso stereoisomer of hydrobenzoin. The second possible outcome is when we produce this stereoisomer of hydrobenzoin along with the enantiomer. This is what is known as a racemic mixture. The third possibility is getting a combination of the racemic mixture as well as the meso compound or the meso stereoisomer. Now, if we take a proton NMR of our product of hydrobenzoin, we would be unable to tell what stereoisomer we have. And this is because proton NMR is unable to distinguish between enantiomers. So the stereoisomers formed here would all have the same proton NMR spectrum. However, proton NMR can distinguish between diastereomers. So a way to overcome this issue is to react hydrobenzoin with acetone and ferric chloride to form the acetonide of hydrobenzoin, and the acetonide is simply a cyclic ketal. When we do that reaction, we would get two possible outcomes. We can either get the cis acetonide or we can get the trans acetonide. Depending on what the proton NMR spectrum shows will determine which stereoisomer was produced in this reduction reaction of benzil to form hydrobenzoin. As you may have noticed, I've boxed in a couple of these structures here in the reaction scheme. Later on, I will explain why I have done this, but before we get to that, let's go over the first part of the reaction, which is the benzoin condensation of benzaldehyde. Before we begin our reaction, we first need to prepare our catalyst, thiamine hydrochloride. So this right here is the molecule of thiamine hydrochloride. And what we're going to do is we react it with a hydroxide anion, and what the hydroxide anion does is the lone pair will grab this proton right here, break the bond, and the lone pairs go onto this carbon. The result of that reaction is this vinylic carbanion. This will act as our catalyst when we react it with benzaldehyde. Now that we've gone ahead and prepared our thiamine catalyst, we react it with benzaldehyde. The lone pairs on the thiamine is going to attack the carbonyl carbon and break this pi bond, and these pi electrons are going to be placed onto this oxygen. The result of that reaction creates this alkoxide anion intermediate. We then react the alkoxide ion with water. The lone pairs on this oxygen will grab the proton on water and will break the bond between the hydrogen and the oxygen. The lone pairs get placed onto this oxygen here. The result of that step produces an alcohol. And now what we have here is this tetrahedral intermediate. And because of the presence of this thiamine group, the molecule now becomes more susceptible to a deprotonation from this hydroxide right here. So hydroxide grabs this proton, we break the bond, the electrons get put onto this carbon right here. This results in the formation of a carbanion. And what's special about this carbanion is that it's resonance stabilized. Normally, carbanions are not that stable because carbon does not like to hold the negative charge. But because it can delocalize the electron density, this becomes a very stable carbanion. What happens is the lone pairs will form a double bond and the pi electrons here will move onto the nitrogen. This creates our second resonance structure. The formation of this resonance stabilized intermediate explains why this proton is acidic, and this only occurs because of the presence of this thiamine group. Next, we take the carbanion that we had just formed and we react it with another mole of benzaldehyde. The electrons here on the carbon will attack the carbonyl carbon, break the double bond, and the electrons are placed onto this oxygen. This results in the formation of another alkoxide anion intermediate. But notice here now that this carbon and this carbon have now formed a bond with one another. So now we've successfully linked the two benzaldehyde molecules. Next, we get an intramolecular proton transfer. So the lone pairs on this oxygen grab that hydrogen, and the bond here breaks, and the bond here breaks, and the electrons are placed onto this oxygen. The transferring of this proton is fairly stable due to this 5-member ring that forms during the transition state during the proton transfer. Next, the lone pairs on this oxygen reform our pi bond, and we expel our thiamine as our leaving group. This regenerates our thiamine catalyst, which can go ahead and react with more benzaldehyde molecules and form our desired product, benzoin. Now, normally, this type of condensation reaction uses cyanide. However, cyanide is a very poisonous and very dangerous reagent to use in the laboratory. Instead, we opt for the more safer reagent, thiamine hydrochloride. Thiamine hydrochloride functions the same way as cyanide in that upon attaching it to benzaldehyde, it decreases the pKa value of that proton on benzaldehyde and allowing us to form benzoin. The next step in the reaction is to oxidize the alcohol group into a carbonyl, and we do this by reacting benzoin with nitric acid. The lone pairs on the alcohol form a bond with the nitrogen, and we break this double bond right here. The result of this is this intermediate right here. Next, an intramolecular proton transfer will occur. The protons on this oxygen here will grab this proton, and the bond between oxygen and the hydrogen will break, and the lone pairs get placed back onto this oxygen. The next step of the reaction is a hydride shift. Remember that this carbon right here still has a hydrogen attached to it. What happens is the electrons form a double bond right here, and we expel this group right here. This results in the formation of benzil, and this group that we had just kicked out from the previous step gets converted into NO2 gas. So when you do this procedure experimentally in the lab, you want to make sure that you direct the NO2 gas away from you by having a well-ventilated area. The next step in the reaction is a reduction of benzil. So we react this molecule with sodium borohydride. Now the first part of the reduction involves this hydride attacking one of the carbonyls. At this moment, when the reduction occurs, the hydride is not selective for any of these carbonyl carbons. So it could attack this carbonyl or this carbonyl. It doesn't really matter at this point. Now, typically when you see this kind of reaction, the first part is the hydride attacking the carbonyl carbon and breaking the double bond, and the pi electrons get pushed onto this oxygen right here. However, what actually happens is the double bond will form a bond with this boron atom right here, and the result of that is this intermediate. Now remember that this molecule is actually three-dimensional, so I've gone ahead and drawn these wedged and dashed lines to indicate the three-dimensionality of the molecule. Also remember that single bonds can rotate. This is important because after we perform this first reduction, this will cause the molecule to rotate along this single bond to form a stable three-dimensional conformation. And remember, we can view that type of conformation using a Newman projection. In this case, we will be looking at this molecule down this axis, So what I've gone ahead and done here is converted this projection of the molecule into a Newman projection. And as you can see right here, you have these bulky phenyl groups that create steric hindrance for this molecule. So what's going to happen is it's going to form multiple conformations until it reaches the most stable one. There are actually 3 different conformations that can be formed after the first reduction. So we can get conformation A, conformation B, or conformation C. As you can see right here, the molecule is going to arrange itself in the most stable Newman projection, and the most stable conformation is the third structure right here, conformation C. And this is because the bulky phenyl groups are furthest away from each other, so this has the least amount of steric hindrance or torsional strain. So now that we've figured out that this is the most stable conformation that will form, the second reduction of this carbonyl will occur, and it will occur where this hydride will attack the carbonyl carbon, and the pi electrons right here will form a bond with this boron, just as we've seen in the first reduction. The result of that reduction forms this intermediate right here, and as you can see now, the phenyl groups are now opposite one another. Sometimes it's a bit difficult to visualize what the molecule looks like in this Newman projection. So what I've gone ahead and done here is converted the Newman projection into a Haworth projection. Now the last step to complete this reduction is to react this molecule with water. This will get rid of the BH2 group and fully convert the carbonyls into hydroxyl groups. Now as you can see here, the OHs are in the cis position, and the stereoisomer that's formed here is the meso stereoisomer. Now the meso stereoisomer is a special type of stereoisomer where the molecule can have 2 or more chirality centers, but its mirror image is actually superimposable with itself. So if we convert this projection into our typical bond line structure, we would see that this is actually an achiral molecule, because if we take the mirror image of this hydrobenzoin molecule, you would see that the mirror image, if we were to flip it, is actually the same. So they can stack on top of each other or they superimpose on one another. So therefore it's not chiral, it is achiral. And this is a special type of achiral molecule called the meso compound or a meso stereoisomer. So just as a quick recap, when we react benzil with sodium borohydride, this will produce the meso stereoisomer. So this shows that this reduction reaction is stereoselective because it produces a specific type of stereoisomer. Now the final step of the reaction is to create the acetonide of hydrobenzoin. Recall that we're unable to take a proton NMR of hydrobenzoin directly because it comes in different stereoisomers. And since the proton NMR spectrum of hydrobenzoin and its different stereoisomers are the same spectrum, we would be unable to tell if we for sure have the meso compound. So we have to take it one step further and synthesize the acetonide of hydrobenzoin. We start off by reacting acetone with ferric chloride. Ferric chloride is a really good Lewis acid, and the reason why it's a good Lewis acid is because it can accept lone pairs, because this iron molecule has an empty p orbital. Acetone in this scenario functions as a Lewis base and will donate its electrons to the iron, and what happens next is we kick out one of these chlorines. The result of that step is this intermediate right here. Next we take the meso stereoisomer of hydrobenzoin and we react it with the intermediate. So the lone pairs on the hydroxide will go and attack the carbonyl carbon right here. The pi bond breaks and the electrons are placed onto the oxygen. The result of that step is this intermediate right here. Next, an intramolecular proton transfer will occur. The lone pairs on this oxygen grab this proton and we break the bond and we push the electrons onto this oxygen. Next, the lone pairs on this oxygen will form a double bond here, and we expel this as our leaving group. This will result in this intermediate right here. This will result in this intermediate right here. And notice that this portion of the molecule has rotated along this single bond right here. And you can see already that the phenyl groups are in the cis position. Next, the lone pair on this hydroxide will come in and attack the carbonyl carbon and push the pi electrons onto this oxygen. This results in the cis acetonide, and the reason it's cis is because these phenyl groups, or you can conversely think that the hydrogens right here are in the cis position. As you can see here, the cis acetonide will only form if the meso stereoisomer of hydrobenzoin was present after the reduction reaction of benzil. Next, let's analyze the protons of the cis acetonide so that we have a better idea of what to expect on our NMR spectrum. We're going to go ahead and ignore the protons on the phenyl rings, because when we look at our NMR spectrum and we see signals appearing in the aromatic region, that will already confirm that we have those protons. Instead, what we're going to focus on are these protons right here. Let's start off with this proton. We see that this proton is surrounded by an oxygen, another proton, and a phenyl ring. But what about this proton right here? Is it in the same chemical environment, or are they in a different chemical environment? Well, let's take a look. This B proton is surrounded by the A proton, another oxygen, and a phenyl ring. So this shows that the A and B proton are actually chemically equivalent. So we would expect to see one signal appear in our NMR spectrum. I will denote this signal for these two protons with the letter A, and also we would expect to see a singlet because this signal has no other neighboring protons nearby. Now, what about these methyl protons? Let's go ahead and analyze these. We have to ask ourselves if this, so we already know that these three hydrogens are attached to this carbon right here. So these three hydrogens are chemically equivalent, so we would expect to see one signal appear for the three of those protons. But now we have to ask ourselves if these methyl protons are in a different chemical environment as these methyl protons. Now, if we take a look at what's neighboring these methyl protons, we see that we have this oxygen and we have these two phenyl rings. Let's take a look at these methyl protons. We see that there's an oxygen, another oxygen, we have this proton, and we have the phenyl rings. So it doesn't seem like they're any different. So are they chemically equivalent? Well, they're not. As you can see by this Haworth projection, these methyl protons, I'll label it B, so these B protons are much closer to the phenyl rings. I'll label this C. These protons, denoted by the letter C, are actually further away from the phenyl ring. So in fact, these B methyl protons are in a different chemical environment compared to the C methyl protons. So this we would expect to see a singlet, and we would also expect to see a singlet as well. So all in all, we expect to see three unique signals in our proton NMR spectrum. Now, you might contradict that and say, well, what about the phenyl protons? Of course, we're going to see signals for these, but these are the more important ones right here, what I labeled A, B, and C. This will for sure indicate if we do have the cis acetonide. Now, if we take a look at our NMR spectrum of the cis acetonide, we in fact do have our molecule. You can see right here that this signal, and this signal correspond to the protons that we had just identified for the Haworth projection. We see that these signals correspond to the protons that we had just identified. This signal right here represents the A protons that we identified. This signal right here represents the B protons because it's much closer to the phenyl ring, so the effect of diamagnetic anisotropy is of greater impact on those protons, so that de-shields those protons more, so we see it having a higher ppm value. And lastly, this signal right here indicates the C protons that we identified from our previous structure. So the proton NMR spectrum does in fact confirm we have our cis acetonide. However, the spectrum is not entirely clean. We can go ahead and ignore this right here because this is simply solvent, and we can ignore this large peak right here because this is another solvent peak. This peak, on the other hand, is something to take note of. Now, what's special about this signal right here is that it's actually the trans acetonide. Now, you might be wondering, how can the trans acetonide appear as an impurity in our proton NMR spectrum? After all, when we perform the reduction of benzil with sodium borohydride, that should only produce the meso stereoisomer. So this means that somewhere along our reaction, there was a potential for the trans acetonide to have formed. Let's try analyzing the reaction scheme once more to see if we can pinpoint where exactly the formation of the trans acetonide could have occurred. So we start off here with our benzoin condensation, and this forms benzoin after we react benzaldehyde with thiamine hydrochloride. So no problems there. Next, if we take benzoin and we react it with nitric acid, this produces benzil. Now this step right here is actually where the trans acetonide can actually form, and I'll explain how. Recall that when you do procedures experimentally, you don't get 100% of your product. Some of the starting material might not have reacted with the reagents. So what if some benzoin was left behind? Well, as you can see here following this arrow, when benzoin reacts with sodium borohydride, it can actually produce a different stereoisomer of hydrobenzoin. And in fact, it produces the racemic mixture of hydrobenzoin, so we get both enantiomers of hydrobenzoin. The meso compound does not form in this scenario because it will only form when you react benzil with sodium borohydride, because it's a stereoselective reaction. Because it's stereoselective, it isn't possible for the mesostereoisomer to form between the reaction of sodium borohydride and benzoin. So afterwards, we would react the product here, so we take the racemic mixture of hydrobenzoin, react it with acetone and ferric chloride, and this will produce the trans acetonide. Let's go ahead and look at the mechanism to see how this all forms. So this is the reduction of benzoin using sodium borohydride as our reducing agent. Recall here that benzoin has a chirality center, so this must mean it has an R and an S enantiomer. So the reduction reaction occurs as you normally expect it to. Now remember, the hydride can attack this carbonyl carbon from the top or from the bottom, because remember that the hybridization of this carbon right here is sp2. sp2 indicates a trigonal planar geometry of the molecule. So this is a flat structure. So the hydride can attack from the top of the double bond, or it can attack from below. What this creates is a new chirality center, as you can see right here. So now this intermediate has two chirality centers, and if we follow the 2 to the nth power rule, this means that we get 4 different stereoisomers. But you have to remember that 2 of these stereoisomers are going to be the same. These 2 right here are enantiomers. However, these 2 stereoisomers are the same thing because this is the meso form, because this is the meso stereoisomer, so these are identical. So the result of the first reduction is the alkoxide anion intermediate. What we do next is we add water, and this will protonate the alkoxide and convert it into an alcohol. The structures that I've boxed in are enantiomers, and remember that this is the product of the reduction of benzoin, because in order for the meso compound to form, you would have to perform the stereoselective reaction between benzil and sodium borohydride. Now let's see what happens when we form the acetonide of the racemic mixture. Once again, the lone pairs make a bond with this iron atom right here. We kick out the chlorine. The lone pairs on this hydroxide attack the carbonyl carbon, and we push the pi electrons onto this oxygen. We perform a intramolecular proton transfer. Afterwards, we form a double bond, and we expel this as our leaving group. This part of the molecule is going to rotate along this single bond to get this structure right here. The lone pair will attack the carbonyl carbon, and the pi electrons break and get pushed onto this oxygen, and we form the trans acetonide. So when we form the acetonide of the racemic mixture, we get the trans diastereomer instead of the cis acetonide, which forms when we have the meso stereoisomer. Now let's analyze the protons of the trans acetonide to see what we would expect in a proton NMR spectrum. These two protons here share the same chemical environment, so therefore we would expect to see one signal for the two of these protons, and we would also expect to see a singlet because there are no neighboring protons nearby this signal. Now the methyl protons are also in the same chemical environment. We would expect to see one signal for these protons as well, and we would expect to see a singlet. And of course, I'm disregarding the phenyl protons because all we have to do is take a look at the aromatic region. If we see that there are peaks that have formed, it will confirm the phenyl protons, but we're not going to worry ourselves over that. We want to focus more on the A and the B signals that will appear on the spectrum. So how is this different from the cis acetonide? Well, this time we would expect to see two unique signals. However, when we analyze the proton NMR spectrum, notice that these protons are going to have the same chemical shift. So they're going to overlap on one another. The chemical shift is going to be the exact same. However, the chemical shift for this singlet is going to be somewhere different compared to the singlets for the methyl protons of the cis acetonide. And as we can see, the arrow where I pointed to earlier is the signal for the methyl protons of the trans acetonide. And this peak right here is the one that overlaps with the cis acetonide. So as we can see here, we do see the two peaks that we expected to observe. So therefore, we found that this signal is indeed the trans acetonide impurity. So just to recap the entire reaction, we take benzaldehyde and we perform a thiamine hydrochloride catalyzed benzoin condensation. This forms benzoin. We oxidize benzoin with nitric acid to form benzil. We reduce benzil with sodium borohydride to produce the meso stereoisomer, and this is because the mechanism shows that the reduction reaction is stereoselective, and this is because when you do the first reduction reaction, it will cause the molecule to rotate and adopt the most stable three-dimensional conformation, and we've seen that with our Newman projections. Afterwards, we form the acetonide, and because we had our mesostereoisomer, it forms the cis acetonide, and we form the acetonide so that we can analyze it using proton NMR. However, if your proton NMR shows some impurity signals, this can be because of the trans acetonide being present inside your sample. And this can occur in this step right here when we try to convert benzoin into benzil. Sometimes not all of this will be 100% converted into our product right here. So we'll have some leftover starting material, and the leftover benzoin can react with sodium borohydride to form the racemic mixture of hydrobenzoin. When we react the racemic mixture with ferric chloride and acetone, we get the trans-acetonide. So I hope you found this video insightful, and I hope you've gained a clearer understanding of the synthesis of hydrobenzoin. Thank you for watching.